Okay, so let's start. We have one more lecture. So we have one more lecture today, and then you have another one on biofouling, and then we'll pick it up tomorrow. And then they will get the solution sometime tonight, right? Okay, so we were talking about virus filtration, and let me just quickly recap. The idea is that we want to validate removal of viruses. There are two types of viruses, retrovirus and parvovirus. At least these are the two main types. And so there are models for these viruses, okay? And so there are many unit operations. Actually, let's start from that point. There are many unit operations that could be used to validate virus clearance. So now that we have an idea of what these all are, let's talk about them. Let's start from that point. Okay, so if you imagine you're coming from the purification train, and we talked about some of this on, I don't know, yesterday or the day before. So some of the aims today, they're still not validated, but the aims are to see if we can remove virus just before this, right, when we have the bioreactor harvesting step. And 3M and other companies are coming out with depth filters that do show some clearance of virus, and they have been validated, but I'm not sure that there are any manufacturing facilities using it still, okay? So remember, when you come up with a new thing like this, firstly the company is validated, but then they have to help you, if you're designing a new processing scheme, to show that you get virus clearance there and that it's acceptable to the regulatory authorities. It is very rarely that you would take a new unit operation like this and put it into an existing scheme. Because if you did, you'd have to revalidate the whole thing, and it's usually not worth it, okay? So, what happened? Is it okay? All right. So, basically, that's one aim, that's here, okay? But in existing work, currently, there is definitely a virus clearance step here, pH adjustment vessel, elute hold vessel. So typically, you elute from the capture step, okay? So this is a bind elute chromatography or adsorption. You bind, you elute your product of interest, and then you adjust the pH. So there is virus, but that's by inactivation, not clearance. Remember I said you have to show virus clearance by inactivation or removal, right? So this is by inactivation. And this only works for retroviruses, okay? Parvoviruses you cannot inactivate that way. You can't adjust the pH enough, and you will damage the protein, okay? So you may get about 3, 4, 5 logs, something like this from this step. You can't validate more than that. And then typically one of these polishing steps, okay, one of these will be used to also validate virus clearance. And that's what membrane adsorbers are all about, and I'll talk about that hopefully later today, but definitely by tomorrow we'll finish that. So that's another virus clearance step. And then the third one that's always almost used is this virus filter, and that's what we were talking about, okay? So those are the main virus clearance steps that are used. Now, this is not to say that people aren't looking at other ways to try and validate virus clearance. There is a thought that can you use precipitation, okay? That would be an orthogonal process. There is no precipitation in this whole thing. So the mode of action of precipitation is different, right? You can say that depth filtration has a different mode of action to tangential flow filtration, or if you like, dead-end normal flow filtration, because depth filtration is not using a size exclusion membrane, you're binding all over the surface. So because of that, the regulatory authorities would allow that, okay? There's going to be a big debate as to whether that can be allowed together with one of these adsorptive chromatography steps, because that's both absorption, but that's something that's to be seen, whether they will allow that, okay? So that's another thing to bear in mind. So like this, we have the virus clearance step. So we were talking about virus filtration, okay? And so let's get back to where we were up to. So I had made it to here, protein capacity. So let's start from this point. Okay, so optimizing involves, this is very important. So if you are designing a virus filtration process, really what you care about is, firstly, you have to have your virus clearance. So every manufacturer sells a virus filter and says it's rated for 5 log, 4 log, whatever, okay? If you can't get that clearance, it's useless, okay? So you have to be able to get the expected virus clearance. You want high flux, because the way you design this is you design it so that you can complete it all in one shift. So it should not take more than 6 hours. If it's going to take more than 6 hours, then there's a problem, okay? So you want high flux. And then you also want high capacity because you want to minimize your membrane surface area. These virus filters are quite expensive. So if you look at the cost of, say, an RO membrane and multiply it by several orders of magnitude, you'll come up with the cost of a virus filtration membrane. So the tiny ones we use in our work, which are probably around, say, 10 square centimeters, that's very small, okay? They are typically selling for close to $1,000, okay? So they're quite expensive. Now, why are they expensive? There are many reasons. One reason is that they have to be validated. So when you send an RO membrane, like they sell it, and if it doesn't work, you might go back to the manufacturer and get a new one. You can't do that here. It has to work. Otherwise, the manufacturer is in deep trouble because you're going to go and charge them for all the protein you lost, which could be millions of dollars. It's almost as strict as a biomedical device, which we'll talk about on Friday. That's one reason. The other reason, it's made to much higher tolerance. You can't let even one virus particle go. So the manufacturing process is more expensive. And then the third reason is it's a very high margin because people make a lot of money on when you make these sort of drugs. So all of this adds into it. So they are expensive, okay? Existing purification trains, this is the important point. In existing train, you probably find these two different virus filters for parvo and retrovirus. Modern ones, and when I say modern, I think this goes back maybe about 4 or 5 years. Anything built after that, you probably only have parvovirus filters that have now been validated to remove both viruses. So again, I mentioned this earlier. It's an interesting point. So it seems logical and obvious if you're using size exclusion that if you design a filter to exclude parvovirus, it will remove the larger retrovirus, right? That seems obvious. Now, the reason it started off the other way is that it's easier to build a filter to remove retrovirus than parvovirus because parvovirus is very small, and so you're going to have problems with product recovery, right, because of this. So that's why they were designed in this order. And so people then put them in the manufacturing train. But then in order to get the regulatory authority in the US and anywhere else to approve just using a parvovirus filter, people had to do a lot of studies. This is studies with mixed viruses, with retrovirus and parvovirus, retrovirus sequentially with parvovirus, parvovirus sequentially with retrovirus, to prove that the fact that you were catching parvovirus didn't affect your retrovirus removal and vice versa. So it wasn't such an easy thing. It took some work and quite a lot of money, okay? And finally, protein capacity is very important because I told you that the product by this stage, well, firstly, you want to have high recovery. Typically, if there's a process that's less than 90% recovery in the biotech industry, that's really not viable, okay? You may use it if you have no alternative, but you want 90% recovery at least. But virus filtration is at the end of the purification train, and so you want much higher recoveries because your product is almost made, right? It's almost pure, so you can't afford to lose it. So protein capacity studies for virus filter applications are the same as the dead-end sterile filtration, but ensuring a good capacity is very important. Important to perform development studies using the same pre-filter. We talked about that. I also mentioned that these pre-filters are very, very complex now because they're designed to remove product aggregates or potential contaminants, okay? And so that's a lot of our research is focused on that. The feed stream I mentioned must be representative so that your virus must be very pure. Otherwise, you're going to get effects that are not real, right? They're not represented in the manufacturing process. And finally, of course, we have a factor of safety for lot-to-lot variability. Okay, moving on. Let me change this now. So this is, I got up to that point. Let's move on now. So virus filtration studies are performed using small-scale discs, self-contained discs, 13 to 47 millimeters in diameter. In the case of the Asahi Planova, which we said were dead-end hollow fiber modules, I said their surface about 10 square centimeters. I should point out this. The Asahi hollow fiber modules are interesting. So they're hollow fibers. They're regenerative cellulose hollow fibers designed like a normal module. So you have a feed port, a retentate port, and you actually have 2 permeate ports, but you run it with the permeate ports closed, okay? But you do have them, so they make them with the permeate port. That is sometimes useful when you're initially flushing and so forth, but they run in dead-end mode, okay? So I'm sorry, what am I saying? You run it with the retentate port closed, okay? So it's in dead-end mode. And one of these permeate ports is closed, okay? All process parameters are scaled down in a linear fashion and should represent worst-case conditions with regard to virus clearance. So linear scaling is important, okay? You assume that everything scales linearly. If it's not linear, it becomes very complicated. That's one thing. The worst-case scenario simply means 2 things. You're using parvovirus, which is this like MVM, so it's 20, these particles are 20 nanometers approximately, 18 to 22 nanometers. You use the smallest virus that is a model virus, because then the thought is if you clear that virus, anything else will be cleared to a higher level, right? That's the idea. Today we use this parvovirus and it still remains the best model. However, in the future, if people discover other sorts of viruses that potentially could contaminate these drugs, and they happen to be more easy to transmit, then we'll have to change the model, okay? So that's how a model comes up, okay? And then worst case conditions, mean, may, square, you have your process hold steps, all these things that could lead to enhanced virus passage through the membrane, okay? And we discussed hold steps, right? When you're changing buffer to flush out the material, you can have back migration of the virus and so forth. Important to process large amount of feed stream per surface area compared with industrial scale down process design to ensure validation under worst case conditions. So all that means is that we scale this based on the liter per meter squared of membrane surface area. This is the amount you're going to pass through the virus filter per membrane surface area. So you have your manufacturing facility, you have 10,000 liters, whatever it is, you know how much, or you determine how much membrane surface area you want, right? And then you scale down. So your small scale test using these 10 centimeter squares, typically we are using about 300 ml of feed, but that is scaled down from manufacturing levels, okay? So everything has to scale, okay? A virus clearance should be measured in several fractions. That's an important thing. So when we're doing these small scale studies, so we have our feed coming here, we are collecting our permeate, this is closed, this is in dead end mode. So we may collect the first, I said we have say 350 ml, we may collect the first 10 ml, and then the next 10 ml, and the next, and the next, and the next, and so forth, okay? And then we'd work out the virus titer in each of these fractions. So you want to see at what point you start to get breakthrough, okay? That's very important. And as I said, virus titer is tricky to determine. The absolute method is infectivity assay, which you have to do anyway, finally, to get approval for the process. But before that, you could use PCR assays and others, but they are not as accurate. So people typically still want to go back to these infectivity assays, and they take a long time to do. Virus clearance can decrease. This is a very important point. Virus clearance can decrease with extent of membrane fouling, reduction in permeability for parvo-type virus filters. So that seems counterintuitive, right? So you're saying as you see a decrease in flux or a decrease in permeability, you're getting more fouling of the membrane. So you would think if you got more fouling of the membrane, right, that you actually would get better clearance of the virus because you have fouled the membrane. But actually often what is seen is quite the reverse, okay? Why? Well, the thought is that as the membrane fouls, you're starting to get more and more of the feed that has to go through the larger pores, all right? So the smaller pores have got blocked by product variants. And so therefore, there's more of a tendency or the possibility for the virus to go through these. That is one hypothesis, okay? And then that seems to work especially for regenerative cellulose membranes. For some of the other membranes, it's not so clear, all right? But this effect is not seen for these larger pore membranes, which is also rather interesting. So the larger retrovirals, don't see it. People are not studying that much these days because they're not used much anymore, but that was not seen that much there. But I should say that this is a very interesting area because it also depends so much on what sort of product variants are fouling the membrane. It depends very much on the mechanical properties of the membrane. So regenerative cellulose membranes are actually more, they're not as rigid as the polyether sulfone. And so you can see that under pressure, you might get slight variations in the pore size. And you're talking about, you know, very small differences. It's not like protein fractionation. What's next? Okay. Another thing about virus filters is that it's a rather complicated process, the whole thing. So you start off, if you're going to run it in a manufacturing procedure, in lab scale testing, everything, you're supposed to first do an integrity test to ensure proper cartridge insulation and or mitigate risk of having to reprocess the feed should the post-integrity test fail. What that's really telling you is you get a virus filter, you put it in there, you want to make sure if it's in the case of hollow fibers, you have no broken hollow fibers. And it's very important to do this, or if you put a membrane, that there's not something wrong with it, because it's very important to do this, because imagine if you find at the end that the virus filter didn't work, you've got to do the whole thing again or throw out your product, and that could be millions of dollars. So first integrity test is basically to make sure that your membrane is totally wet, because if it's not wet, you won't get liquid going through all of the fibers or the membrane surface area. You can't guarantee that it's going to operate properly, okay? And then you also test it, you do a flux decay test and so forth to make sure that there are no broken fibers. So that's a procedure the manufacturer gives you. Then you run your virus filtration. You do your pause step, you flush out extra protein and do all this. And then you've got to do this post-integrity test is required to ensure that the claimed virus clearance was in fact achieved, since it is not feasible to determine virus content in the filtrate at low levels required for human pharmaceuticals. But the fact is that there may not be any virus there, right? So you can't take a fraction of your product and say, I'm going to test it for virus clearance, because that won't tell you anything. Because there could be virus, there may not be, it may be below level of detection, you just don't know, okay? So what you have to do instead is say, okay, I've finished the virus filtration, this is a one-time use device, I need to now test this and make sure that actually it rejected virus. Each of the membrane manufacturers has their own test. So one test that Asahi does is that they use gold particles for their regenerate cellulose membranes. So you challenge it with gold particles, okay, and you look at how many gold particles are retained, how many pass. And Asahi has come up with a correlation to relate that to the virus clearance you would expect, okay? And they have validated that, and in the US, for example, the FDA accepts that this is correct. Therefore, if you do this gold particle test and you get what you want, fine, it worked. Millipore has developed a test based on, and actually Asahi is moving away from gold particles. It turns out that gold particles are just very difficult to do. So these membrane manufacturers tend to use basically a liquid, liquid intrusion test. So they're using two different liquids. And based on that and how much goes through, you can tell whether this, with different surface tensions, whether in fact you got the clearance you would require. These are all destructive tests, which means once you do the test, you can't use the filter again, okay? But that's fine, you're going to throw it away anyway, okay? But this is a very important part, because if you fail this test, which could happen, you've lost your product or you're going to reprocess it. And reprocessing is not easy, okay? Finally, the filter manufacturer must also correlate the integrity test with the virus clearance. Okay, my final slide in this area then is I've tried to in every lecture talk about where we are heading in the future. So the final slide talks about future directions. So this is more of a thought exercise. So I told we want to move towards continuous biomanufacturing, right? So how would you run a virus filtration experiment or design an experiment so that you could do this for continuous biomanufacturing? So this means, just for argument's sake, we're going to run for 1 month, okay, as opposed to 6 hours. So our product is coming through, and you're going to put it through this virus filter for a month. Here are some of the challenges. So how would you validate this? Because, you know, typically you run it, you do a destructive test, and you work out how much you removed and so forth. Now, if during this month this filter fails, how are you going to tell this? Because you don't know, right? And if it does fail, then you have a big variation in product quality. This is a major issue, okay? The second thing is, if you're trying to do a validation test in the lab, okay, so in a lab test we spike with virus to check and scale down experiment. How would you run a scale down experiment? Because, you know, we can't, so we have a stock feed with virus in it, and we typically then do the filtration. But these virus are not going to actually be active and stable for 1 month in the buffer, okay? Parvovirus, maybe if you're lucky, 3 days or so. Retrovirus, very short, okay? Maybe 6 hours is maximum. Parvovirus, maybe 3 days. Depends on the pH, buffer conditions, temperature. But certainly not 1 month. And remember, the final test you do has to be an infectivity test. So if the virus is no longer viable, then infectivity will be 0. Did you get clearance, or did you just have virus particles that are not viable? It's a major issue. So that's something that actually is a major concern today. If you're going to do continuous biomanufacturing, how do you validate this at the lab scale? That's one question. And then how would you run these? Another thing here that's interesting, I told you want to run at high flux because you want to get everything done in one shift. But for continuous biomanufacturing, you don't need that. Low flux is fine because you're producing the product all the time at a low rate but continuously. Also, virus filters today are used in the constant pressure mode, okay? But if you're doing continuous biomanufacturing, you might want to run on constant flux, low but constant flux. And so we've done quite a lot of work in this area that shows that existing virus filters do not work very well at low flux. And just very briefly, the reason would be this. So at high flux, you go and you get the viruses trapped in the pores and structure. At low flux, there's a possibility that there's Brownian diffusion that could allow them to back migrate and find larger pores to go through, you see. That would be a problem. So there's a lot of work to be done in terms of designing virus filters for continuous biomanufacturing, for looking at constant flux versus constant pressure, and for looking at validation of these devices for continuous biomanufacturing, okay? So I think that's the end of what I want to talk about for virus clearance. And our next lecture, which was to have been the morning lecture, is actually going to be on adsorptive membranes. And it works well this way because adsorptive membranes are also used to remove virus particles. So let me share again. Is it coming? Yes. Okay. So I think this is actually the last. No, we have some lectures tomorrow on future applications or future developments, but this is getting towards the end of our work on protein purification. So what I'm going to talk about now is actually one of these two steps, either this one or this one. So to put it into perspective, these two polishing steps, and the platform would like to have two, maybe you need more. If you can do it with one, that's better. The idea is you've done your capture step here with protein affinity chromatography. You bind your product of interest, you let everything wash through, you recover your product of interest. Now you're going to see, can I polish this? In other words, remove product variants, DNA, host cell proteins, things like this. That's what we're aiming at, okay? So membrane adsorbers were also known as membrane chromatography, okay? And what they do is they use microfiltration membranes or larger as chromatographic support materials. So I think all of you are familiar with especially chemical engineers, should be familiar with something called packed bed adsorption, right? Everybody knows what that is, right? So you take little spherical particles, typically resin particles, you pack them in a bed, right? You run your feed through that, you bind what you want, and then you elute, and you run a packed bed adsorption, right? And typically these particles are porous, so you maximize the surface area because most of the binding occurs in the inside pores. One thing to remember, in the biotech industry, these packed bed processes are called chromatography, okay? So as chemical engineers and as chemists, you would think of chromatography as what? Something where you have differential rates of interaction with the resin, and based on that, you get these peaks separating, right? Right? So, but so-called frontal chromatography. But in the biotech industry, every packed bed process is called chromatography. So you have ion exchange chromatography, hydrophobic interaction chromatography, but really they are packed bed adsorption processes, okay? And the reason for that actually is that it was life scientists and biologists that initially designed these purification trains, and so they weren't as strict in terms of what words that should be used. So membrane adsorbers or membrane chromatography basically says get rid of this idea, and instead what we're going to do is we're going to take a membrane. So imagine our flat sheet membrane, which I'm going to try and show you like this, and it's got pores like this, right? We've got pores here and interconnections, and in these pores we're going to add ligands. Okay, so we do some surface modification and add ligands, And now we're going to pump our feed like this through the pores. We pump it like in microfiltration. And as the feed goes through here, we can absorb species that we want to either bind or remove. Okay, we can do this. And we can elute and do various things. So the idea is that you use membranes as chromatographic support materials that contain functional ligands attached to the inner pore surface throughout the membrane structure to provide a highly selective separation through adsorption binding interactions. Okay. What is used today? We have ion exchange, we have reverse phase, we have hydrophobic interaction. Membranes have all been developed, but basically ion exchange and hydrophobic interaction are the two most common. Okay, they tend to be used routinely in the biotech industry. The others are developing. Okay, affinity binding capacity in membranes tend to be low. Convective flow through the pore reduces mass transfer resistance. A disadvantage in membranes is actually if you took the membrane pore surface and you compare that to a resin particle, you actually will have a lower density of ligands. So in other words, the capacity of this is less than for resins. Okay, that's a disadvantage because you want high capacity. Okay, you want to minimize the volume of these things. The advantage is a fluid mechanics advantage. So if again summarizing this, if you think of what happens, and so everyone who has chemical engineering degrees would know this. If you think of the binding of something to a resin particle, we talk about various mass transfer resistances, right? So you start off by pumping through your column, you've got an external film resistance, you got to get to the particle, then you've got a resistance to go through the pores, then you adsorb, then you desorb, you come out, and so forth. This pore diffusional resistance is quite important because it's quite slow. And what happens is that it means that the dynamic capacity turns to be a function of flow rate. So what do I mean by that? I mean, if I take the resin particles and I put them in a beaker and I add some BSA and I have very, very slow movement to make sure that it remains a nice mixed solution, you will get static binding capacities that give you the maximum coverage. Forget about that. Put them in a column and pump your liquid through. If there isn't enough time for the species, the BSA in this case, to go into the pores and bind, it will flow through and out. Okay? And there are various Darmkohler numbers we talk about in chemical engineering that you can use to determine when convective versus diffusional resistance are important and so forth. But that is very important in the biotech industry because we are not talking about BSA or small species, we're talking about rather large proteins. And they have therefore very low diffusion, pore diffusion coefficients. So it's very easy to basically have what's called early breakthrough. In other words, you get breakthrough of this before you have fully adsorbed everything inside the pores. So you're wasting half of your column. So that's a major disadvantage, but the capacity tends to be lower. That's one of the limitations. I would point out a lot of research we do these days has increased the capacity of these membranes. Important for purification of large biomolecules and viruses that can have significant diffusion limitations in conventional chromatographic media, okay? And these diffusion limitations means you don't get a very effective separation. Membranes adsorbers, this is what's important. They display high flow rates, low pressure drop, and shorter processing times. Well, since I'm pumping through the membrane pores, there's basically no pore diffusional resistance. We have convective flow through the pores, okay? So that leads to what? Higher flow rates, shorter processing times. That's very important because another thing to point out, these proteins do destabilize and you do lose some due to aggregation during each of the purification steps, and especially in chromatographic purification steps, okay? Because they are shear sensitive and so forth. So you want to minimize processing time. Also, the pressure drop is lower because you have a much more open structure, and that also reduces damage to the proteins, okay? So let me summarize that. So, So packed bed chromatography frequently used in the biotechnology industry. Convective flow through the bed is fast for membranes, okay? Pressure, convective through the bed is slow. We'd like to have it fast, convective flow through the pores. The pressure drop tends to be high in resins, but in membranes we can have that low. And pore diffusional resistances are rather high, so it's slow for resin particles, but we'd like to have the opposite. And so if we look at ideal properties, what we would like to do then is have a low pressure drop, high throughput without channeling. Another thing I forgot to mention, packing a small column for lab scale is fine, Packing a huge column, and if you look at these, sometimes they're 1 meter in diameter. It's not easy to pack those, okay? You have to pack them. You're not going to take it and put the resin by hand, okay? It's going to be done mechanically. And if you don't have it done uniformly, you will have what in engineering we call channeling and bypassing of some of the resin particles. Your capacity goes down again. You're wasting them, okay? So we'd like the separation, this is in terms of mass transfer, to be limited by the kinetics of adsorption and not pore diffusion. So if you study this, you have various mass transfer resistances, and we want the kinetics of adsorption to be limiting. Dynamic capacity, we'd like that to be independent of flow rate, and so we come up, that's where the attraction of these absorptive membranes are, or pure membrane chromatography. Okay, so the interesting point about this is that membrane chromatography, as it was called, today they are sold as membrane adsorbers, but it's the same thing, actually goes back to the 1980s, and I actually worked for one of the first companies that tried to commercialize this in the 80s, but it didn't work, it failed. And this is an example of slow commercialization, and the reason was the binding capacities were low, membranes are lower anyway, and secondly, the membranes are not very uniform in pore size, so you ended up then with the same problems you have with resin, faster flow through larger pores, slower flow through other pores. The faster pores bind quickly, reach saturation, you have early breakthrough, okay? So that's one of the reasons why they could never compete with resins, even though there are all these potential advantages, until about maybe 15 years ago, okay, 10 to 15 years ago. And that's due to recent improvements in membrane materials, chemistries, coupled with a better focus on niche applications, generated renewed interest in using membrane chromatography bioseparations. Now, here's something very interesting. I use this word niche applications, and that's important because, again, the biotech industry is a complicated industry. If I come up with a new unit operation and tell you I want to use this, well, the first thing I'll do is get it validated, and that costs a lot of money. So unless you can prove it's going to be significantly better, there is no motivation to do this, right? And you can't really prove it until you've used it, okay? And so that's the problem here. And so the niche application is used in what they call flow-through mode. So usually we bind what we want, right? We let all the contaminants go through, we elute our product. That's fine, but there's a problem there if in fact your capacity limited, because then you might have high concentration of your product and you can't bind it effectively, you need too much resin, right, or too many membranes. But what we want to do here is actually bind the contaminants. So we don't bind the product. We pump through, and as we are pumping through, the contaminants are bound onto the membrane surface, and the product flows through, right? Now, if the contaminants are the minor component, and they are, the virus particles I told you, they may be 10 to the 6, maybe a lot, but it's much less than the number of protein molecules present, right? So this works then, right? You're binding the minor component, and so maybe the capacity is not such an issue here. Another thing, when you bind proteins and release them, there is the risk that you will denature them and affect their properties. So here it doesn't matter, they just flow straight through. So this is the niche application, flow-through mode, polishing operations, removal of contaminants. So viruses, host cell, HCPs, host cell protein, and DNA. So until we started thinking about membrane adsorbers in terms of flow-through mode, they were never competing. But today they are found to be very, very effective for this. So recent studies on ion exchange resins demonstrated presence of exclusion phenomena that reduce mass transfer rate of protein into beads. So that's one thing to note. The phenomena occurs for large proteins such as monoclonal antibodies and using typical resin pores. The effect seems to be due to initial binding of proteins at the pore entrance that results in steric hindrance and charge repulsion of subsequent protein entering the pore. So all of these are limitations for resin columns. Using large pore resins reduces capacity. Of course, you get around it by using larger pores and you drop in capacity. These effects are also observed for smaller protein fragments such as antigen binding fragments 44 kilodalton for smaller resin pores. So these are some of the disadvantages for resins. It's not to say that you can't get this with membranes as well. Obviously, with membranes also, you can get binding at the pore entrance, but it's not so significant because you're pumping by convective flow, excuse me, through the membranes. Okay, how are we doing for time? Okay, studies of membrane chromatography have shown that the exclusion phenomena also exist with membrane media. But as I said, since we are using convective flow, sometimes it is not so serious. Extrusion phenomena has been directly observed in beads using confocal microscopy. If you are interested, there are some very interesting papers in that area. Since pores are used for convective flow through a membrane media, and extrusion phenomena may exist due to either surface extrusion or flat binding or pore extrusion from membrane surface modification. This is just saying that this same effect can be seen, but actually it is less significant for membranes. A range of membrane-based materials have been used. They have to be modified by surface modification. So Paul Corporation makes so-called Mustang Q and S. So just remember, Q means quaternary amine, okay? So it's an anion exchange membrane. It's got a positively charged group there. So it's exchanging anions. And S means sulfonic acid groups, okay? So it's a cation exchange membrane, okay? And they use flat sheet polyether sulfone modified with quaternary aminosulfonic groups. Sartobind Q, this is sartorius, has a whole range of membranes. Sartobind Q membranes of sartorius are made from stabilized regenerated cellulose. Okay, so here is to summarize in a pictorial way what I was telling you. So here is a resin particle, right? And you have flow outside the resin particle. You have convective flow outside. Here are the internal pores. You're going to have a bulk convection. You have this film resistance outside the particle, and you have a pore diffusional resistance because your protein, whatever it has to diffuse into the pores and bind. Here is the other way with the membrane. With the membrane, you're going to pump through the pores, so there's no more of this pore diffusion. You do have a slight film resistance on the surface, but that is why membranes are attractive. Okay. Okay, how do we make these membrane devices? Well, we can have stacked discs, and so the stacked discs are an interesting idea because what happens is you can take one disc and put a second disc and a third and a fourth and a fifth like this and stack them up. Why is that useful? Well, we know that membranes have a pore size distribution, right? But we actually want a uniform pore size, as I showed you in that diagram. So as you stack them, you tend to get a more uniform pore size, right? Because you have bigger pores, smaller pores, but they should be averaging out to a more uniform one because you want the same flow rate through all these pores. You want the residence time to be the same. If you don't have the same residence time, you will have early breakthrough effects. So it's achieved by multiple layers, provides more uniform flow distribution with defects in one layer compensated by flow through subsequent layers. We can have pleated cartridges for membrane chromatography, similar to those used for normal flow filtration. That's another way this could be done. And the equipment modules available with membrane media volumes ranging from 0.35 ml to 5 liters, covering a range of flow rates of 3.5 ml per minute to 50 liters per minute. So that's a huge range, okay? Just one thing to remember, the reason they say media volumes ranging from 0.35 ml to 5 liters, we don't typically talk in say, reverse osmosis or membrane technology about the volume of the membrane, do we? We usually talk about surface area. But in this work, we talk about volume simply because in packed bed chromatography, people typically talk about the volume of their resin. So it's just in order to compare things. The same nomenclature and terms are used, okay? Okay, so flow through, which is what I just mentioned. So growing use of flow through applications in which buffer conditions are set to enable binding of impurities, right? Binding of impurities, while the product of interest will allow to flow through the media. These applications leverage high linear velocities possible with membranes while maintaining small media volume, generally required for binding small amounts, that's important, small amounts of impurities in the polishing step. So the lower capacity is not so critical. What are we removing? DNA, virus, endotoxin, and host cell proteins is comparable to resins where clearance of host cell proteins may require low conductivities to achieve the same level of removal. Due to small media requirements for flow-through applications, it's possible to use membrane chromatography as single-use devices. So that's the other point. When you're using resins, you typically have to regenerate them. And as I said, that's rather complex, especially when you're removing contaminants. You've got to validate that they've been taken out. In the case of membrane chromatography absorbers, they are one-time use. You use them, so they're designed like this, okay? And so this is a major advantage. So we eliminate the need for reuse studies at small scale and for regeneration and sanitization at large scale. Okay. Oops, I'm sorry. Okay, bind and elute. So membrane adsorbers have found use in bind and elute applications for large solids such as DNA, RNA, and viruses. So this actually moves on to some of the emerging methods and virus purification, which we'll talk about maybe tomorrow. But for protein purification, it is rare to use membrane adsorbers in bind and elute mode. Remember, bind and elute now means that it is important what the capacity is, how many ligands we have there. But that is relatively rare. But it is developing, and so that's an area of development. Most resins have pore sizes that exclude very large solutes, and that is one of the reasons that membrane chromatography is interesting, because if you're excluding the solutes from the pores, you have a very low capacity because you're binding only on the outside. Membrane adsorbers provide competitive DBC's dynamic binding capacity for such applications, where you have large particles and large proteins. Currently, large internal surface area of resins combined with high media packing densities has made it difficult for membranes to compete on bind and elute, as I mentioned, applications for small solutes in terms of dynamic binding capacity, number of process cycles, and economics. But for larger solutes and viruses, there is starting to appear applications. Okay? Significant efforts have been made to develop membrane adsorbers for initial capture step. The initial capture step is a protein A bind and elute. Okay, I don't know of anyone using that commercially, but if you look, you can now find protein A membranes. But I don't know how many are used actually in manufacturing facilities. But certainly for small scale pilot work, they are used now. Use of immobilized hydrogel supported by an inert framework of grafting of polymer brushes onto polypropylene membranes, and use of surface modification. Electrospun membranes is a new area of researchers. I think we discussed that. This is a very interesting idea because they could potentially provide a very low hydrodynamic, very low resistance to flow and high capacities. Protein A affinity membranes are now available, I said here, with similar capacities as protein A resins, but these are typically too expensive for single-use applications. So their use is still minimized. Multimodal membranes are more attractive for single-use capture. So what do I mean by a multimodal membrane? So we talked about ion exchange membranes, and they have cation groups and ion groups, right? Multimodal means that instead of using a quaternary amine, for example, you might use a tertiary or secondary or primary amine, and so on those other H's from the nitrogen, you can replace those with hydrophobic groups, okay? And so you can have hydrophobic interaction and ion exchange interaction. So you're using multiple mechanisms, okay, which would increase your capacity. I should point out you don't gain something for nothing. So multimodal membranes are also problematic because you could actually adsorb some of your product more easily, right? Because now the product may interact by hydrophobic interaction or something. So it's a little tricky. But that's a great area of interest at the moment. So single-use devices, so single-use disposable chromatography systems are particularly attractive to small-scale bioprocessing, production of material for early clinical trials, and for manufacture of products with relatively small annual demand, because it's quite expensive, right, to be able to clean and redo the whole thing. These systems eliminate column packing, which is a major concern, because if you can't pack your column reproducibly, you get poor results. Operation, reducing labor costs and providing more uniform flow properties than can be obtained even with automatic packing systems. We also limit the need for column stripping, clean in place, and regeneration steps, minimize introduction of bioburden, which can be a concern during column packing. And this is just showing a CM of NatriFlow, which is a membrane chromatography media consisting of functionalized polyacrylamide hydrogel supported by a matrix. Matrix actually is a company that was bought up by Millipore, and they make some unique membrane type chromatography resins. So these are the advantages of single use. Basically, you don't need to regenerate, you don't need to clean in place, right? And so this is, and you don't have to dispose of all that cleaning material, whether it be buffer, whatever it is. And so that actually is a significant advantage. Module design. I'm wondering, should we, I think everyone is looking tired. Should we take a break? Because they're going to start their next lecture in about 7 minutes or something, right? So maybe we will pick this up tomorrow. We've got to talk a bit about module design here, and then I was going to talk about hybrid applications and some continuous countercurrent tangential flow chromatography, which is some of the newer developments. And then we'll start tomorrow's lecture, which in the afternoon will focus on emerging therapeutics. So let's stop at that. I think that's a good place to stop. Okay? All right. Thank you. Sure.
So what type of ligands are attached on the membrane for membrane absorption? If it's just increasing the hydrophobicity, everything could potentially absorb because most of the components are they would have a hydrophobic interaction or any cells, all proteins.
Yeah. So the easiest one is adding ion exchange. Okay. So quaternary amine or sulfonic acid or carboxylic groups. That's a weaker one, and then tertiary amines and so forth. There are hydrophobic interactions that are adding phenyl groups and things like this. Typically, they are using the ligands that come from the hydrophobic interaction resin-based chromatography, these same groups. The multimodal ligands tend to be like guanidine and things like this that are being put there, and they have more specific interactions, say with benzene rings and so forth.
Then how would it be eluted out?
Either change in pH or change in salt concentration, either one. Yes, yes. I would say today the multimodal ligands though are still mainly used for flow-through applications. Okay. One of the things is removing aggregates. That's something that you say the aggregates have slightly different hydrophobicities than to the native protein. So that's another one. But the bind and elute applications for membrane adsorbers are very much still developing. There are a few applications coming up, as it said, for small scale processing, but for big scale it's not done yet. It is also coming up for purification of virus particles. There we have a real problem with resins. Okay. All right. So any other questions? Yes.
Yeah. What kind of membranes, like asymmetric, symmetric, or reverse asymmetric membranes are used for?
That's a good question. Symmetric. You want symmetric, large pore microfiltration membranes, or even larger.
So we can use asymmetric also, right, sir? When you use asymmetric,
Yeah.
reverse asymmetric, I guess. So then you are giving a time for the interaction of the, because as particles like impurities are less in concentration.
Yes.
So you give a time for the impurities to bind to the ligands, so that you get a good capacity.
That's a good point, but actually you can also do that by slowing your flow rate. Right? If you lower the flow rate, you can do the same thing. And you will have more control if you use a symmetric membrane. Because what you want to do is this. If you think of it, ideally, an ideal membrane is like a track-etched membrane, okay, which has got very high porosity, which is impossible, very high porosity, and also interconnections. That would be ideal, because if you had exactly the same pore structure repeating, you'd have the same flow rate through all of it. You'd like to have the same resistance, okay? But we can't do that. So we're using phase inversion that has this pore size distribution. But if we make it symmetric, we have more control over those. Asymmetric becomes tricky, that's all. Actually, there are some other interesting ideas. So we once made something called inverse colloidal crystal membranes, where we, if you imagine, take a whole lot of colloids, put them together, so spheres, put them together, let them set in some sort of polymer and etch out the spheres. And then you get actually a very uniform membrane, because what you'll get is something like this. So you'll get something like pores looking like this, right? So these were our original colloids, and you etch them all out, okay? And then you can get interconnections, because something will interact here, so this will do something like this, and you get a connection here and a connection here and so forth. That's totally uniform. So if you can get totally uniform repeating structure, that'll be great. People have tried to do that also by using different sorts of dibond polymers and so forth. But to do that for virus clearance is tricky, because you need rather large pores. For smaller pore sizes, it may be possible. The reason this has not really taken off is that these methods are very nice, but you can't really do that at large scale commercially. It's not possible to scale up very easily. Okay? So yeah, asymmetric could be a way, I agree with you, to lower the flow rate, but actually just lower the rate at which you pump would be easier. Okay? All right? Very good. So I will see you all tomorrow then. And let me turn this off.